Everyone. Hello, everyone. Good morning to all of you. Thanks a lot for joining us today for the first day of the workshop. And we are super happy to have you in this program that we call algal bloom monitoring or monitoring algal bloom. So I think all of you have met us already. If not, then I'll take just 1 minute quickly for all of us to introduce so that, just to refresh your memory. My name is Iqbal Chaudhary, and I am a researcher, and together with this amazing group of people, we have started something called Open Field Collective to run programs like this with amazing teachers like you. And I'll let others speak for quickly just to introduce themselves, and then I'll take over. On my screen, I have Kanika first. So Kanika, can you please go ahead and just say a few words about yourself?
Hello, everyone. I don't think I met anybody because I had not come to the previous meetings, but I'm Kanika, and I'm also a postdoc scientist here in California, in University of California, Berkeley. And I work on imaging technologies to figure out host pathogen interactions. And I'm excited to talk to all of you today about algae and what we do in our workshop.
Next, Ankit, do you want to go?
Hello, everyone. I am Ankit Trivedi. I am a bioinformatic analyst working at University of Maryland School of Medicine. And I've been a part of OFC since the beginning, and I'm really happy to be a part of this workshop as well.
Sayak?
Hi, my name is Sayak Bhattacharya. I'm also a computational biologist. I work at MIT in Boston. Like I'm all this stuff, like ever since OFC started, all the work with algae and microscopes are also very new to me as well. So I have learned a lot throughout this process, and I'm very excited to keep that going.
Ankita.
Hi, I'm Ankita. I am a scientist at National Institutes of Health in Washington, D.C. I'm also the one you interact with in the WhatsApp group. So it's nice to see some of the faces over here. And I'll give it back to Iqbal to start.
Yeah, thank you. So just some housekeeping rules as we are going to go through the presentation. Please keep yourself muted. But if you have any question, feel free to unmute yourself. And also you can turn on the video if you are comfortable. And speak up your mind, like let us know whatever you want to talk about, any language that you prefer. I think most of us are comfortable in Hindi and English and Bengali. So Hindi and English would do, I guess. So let's move on. I think I have said whatever I wanted to say, but regarding the project, so the project, as I said, is called Algal Bloom Monitoring. And it's a citizen science project in the sense that this project will be run by all of us in a huge collaboration. So all of us are in different locations, doing different things, but the idea is we all come together to do one big project as a team. Now, what are the advantages of doing such a project together? Is one thing that we primarily care about is educating our next generation of scientists or engineers or doctors or in general citizens, just to improve the scientific temper in the next generation. So we focus primarily on doing this project with teachers and school children, which you are aware by now. And in order to do that, we have sent a bunch of tools for you to use and learn from. So some of the tools that you have already received, I'm assuming you have already received, are microscope called Foldscope, and some of the components to make a spectroscope, glass slides, ring stickers, and so on. And so in this course of the workshop, we are going to teach you how to use these tools. Some of the things you will use right away, the way the microscope is. And regarding the spectroscope, we will teach you how to build the spectroscope and the physics of spectroscopy, the biology of monitoring environment and algae and all that. And with that knowledge base that we will teach you for about a month, what we will do is we will take that knowledge and try to apply it into a long-term project that will go on for about a year. So that's when you will identify a freshwater body near you, and then go to the water body once a month to collect water sample with your students, and then measure the algal bloom in that water sample, preferably with your students, so that the students learn and you all have ownership of the data that you generate. And then we can all look at the data, that what is going on in your local water body. Okay, that is extremely important, not only from a public health issue, I mean, importance, but also in general, hygiene and health in your local surrounding. Okay, next slide, please. So far we have, let's do a tally of what you have received. And if you have any questions, please feel free to ask the question via the chat box, or you can just say hi and ask your questions. So it is possible that you all have received different kinds of kits, because we were trying to optimize the tools that we have and try to distribute it in the most efficient way. But if you haven't received something, do let us know from the get-go, so that we can make sure that you get it from Abhishek ji. Yes, so here, as you can see, the basic and the most important items that you would need to go through the workshop and also do the project is a fold scope, which is a paper-based microscope, a couple of, cardboard sheets. Are we okay, Ankita? Is there any lag, or everyone?
No, we're good.
Okay,
Yeah.
no, I thought there was a lag from my side. So cardboard sheets, pH paper, transparent cuvettes, which are for holding your water sample, filtering equipment, diffraction grating. So please bear with me. These things may sound very complicated, but once you get used to the idea, you will see that you can do amazing things with these things. Glass slides, printed slits, and so on. Can we go to the next slide, please? Yes, so one thing that I wanted to clarify from the very get-go is that some of you may have received a different version of fold scope. If you have talked to other teachers, then you may, during the workshop that Rafiq and Mo and others conducted, you may have compared your fold scope and found out that some of you have a different version. I just wanted a quick show of hands, or just a confirmation that how many of you have a fold scope that was already assembled. You can speak up, or you can just raise your hand.
Yeah. Hi. I think nobody has, because we all have to assemble. We have the workshop which was conducted at DIET, where we assembled these fold scope by our own. So nobody, I think, no one is there who got the ready-made or the assembled fold scope.
I see. Thank you so much, Rashiji. Now that makes us a little bit.
It's Bharti.
Hi, Bharti.
Yeah, thanks.
Oh, okay. I thought someone else was talking, but my mistake. Yeah. So now that we know that you didn't get the one on the right-hand side, you didn't get the one on the left-hand side, but you got the one at the center, right? So where you had to assemble the fold scope. Now, the next question is, did you all manage to assemble the fold scope?
Yes, of course. The trainers were so good, so good. And it was a very quick bite, and we really thoroughly enjoyed and very much excited to fold it again.
Awesome. That makes us very happy. Okay, I think then we are more or less done. We know that you all got the kits. Can we go one slide back, please? Yeah, here, does anyone have any question about the things they didn't receive or something, like cuvettes, glass slides, printed slits?
Again, it's me. I think we didn't have the pH paper strips, as well as diffracting grating, I'm not sure, and printed slits. Maybe these three are in my queue list, which I don't have any clue about it, because pH strips are definitely not there.
Okay, let me talk to the team in Bulandshahr. Let me find out why. Yes.
सर ये प्रिंटेड स्लाइड्स नहीं है भारती मैम, अलग से जो पेपर दिया गया था एक रैपर था, उसके अंदर सारे थे फिल्टर कोन वगैरह, ट्रांसपेरेंट क्यूबेट्स और ये सब थे, बस प्रिंटेड स्लाइड्स नहीं थी। आप
प्रिंटेड स्लिट्स मैम, प्रिंटेड स्लिट्स और पीएच पेपर्स दोनों ही नहीं है,
और डिफ्रैक्शन ग्रेटिंग भी शायद, राइट?
शायद नहीं है, डिफ्रेक्टिंग ग्रेटिंग भी नहीं है।
हाँ, तो डिफ्रैक्शन ग्रेटिंग काफी एक बड़ा कार्डबोर्ड का टुकड़ा है जो एक ट्रांसपेरेंट पेपर उसमें सैंडविच है, वो आपको दिख जाएगा, अगर नहीं है तो क्लियरली वो नहीं है, वो मतलब छुप नहीं सकता। और प्रिंटेड स्लिट जो है वो काफी छोटा है, लाइक ऐसे 1 सेंटीमीटर बाय 1 सेंटीमीटर का एक छोटा सा काला टुकड़ा है प्लास्टिक का, जिसमें से एक लाइन बना हुआ है ट्रांसपेरेंट। सो मोस्ट लाइकली वो लास्ट डे पे मैंने रफीक से बात करी थी, तो मैं एक बार कंफर्म करता हूँ वो है, बट वो मिल जाएगा, बट देयर इज नो नीड टू वरी बिकॉज वी डोंट नीड इट राइट अवे। द नेक्स्ट 2 सेशंस विल बी ऑन फोल्ड स्कोप एंड मेनी अदर थिंग्स, सो आप लोग चिंता ना करें, वो मिल जाएगा। Okay, I think I'm more or less done with introduction. Ankita, do you want to take over?
Yeah, sure. So, I mean, we thought we will start with the very basic of what we need for the whole program. And one other thing we are going to start with is, talking about microbes and also classification of the living system, which I'm sure most of you are very aware with. And then talk a little bit about the basics of microscopes. And then I think Kanika is going to talk about what we mean by algal bloom and why algal bloom is harmful and why are we doing this project ultimately. So Iqbal, if you move forward. So the classification of living system can be done in different ways, but one of the most famous way how Robert Whittaker did was in 5 classes, starting from the animal one on the left of the picture there, which includes us and other animals like lion over there or fish. Then the other classification system, which we see a lot around, is the plant one. Then the third classification was the fungi, which is he put that classification separately because he could not put that in any other classification. So things like mushroom or, any other kind of fungi which are around in our system has a separate category. So it's not really based on how these living organisms were evolved, but more basis on how the cells were and how the system was working. The other 2 major classification was the protist and the Monera classification. So protist includes most of the algae, which we are going to talk about later on. And then the Monera, which is on the bottom of the picture there, has more of these archaic organisms like bacteria, and he just put that in a completely separate category. So again, this is not a hard and fast rule of classification, and then we will go forward and talk about other ways to classify living system. So if you move forward to the next one, the other thing which is sort of very important to think about is the length scale of life. So if we look at ourselves, which is on the extreme right, we know that we are huge, so we can measure that in terms of some meters, right? So this is something which is visible to our eyes. The other stuff is visible to our eyes are these large plants we see. And similarly, if we go on the left of the scale, the organism starts to become smaller and smaller. So if you look at the bottom, there is a panel which says about the scale there, which goes from meter to millimeter and then to micron. And on the bottom, it says human eyes and magnify. So at some point, our visible eyes are not able to see certain organisms or a certain scale because we are not able to resolve that. So because our eyes also have certain kind of a lens to be able to resolve these organisms. So if you go towards the left, things like animal cell, which eventually we are made of. So if you think about your skin or your body, it's composed of these cells, which are like bricks, which make a building, but you can't really see these bricks by your naked eye. So it's very hard to see that. And that's where, things like, or the instruments like microscope become really important to look at that particular scale. So if you go to the next one, again, thinking about the one easy way to think about length scale of life is to think about your hair. So if you look at the hair, the diameter of the hair, which already, if you look at by your naked eye, is very thin, and the diameter of your hair is somewhere around 70 to 100 micron. So to give you an idea of what a micron is, 1 micron is 1 millionth part of a meter. So you kind of know what a meter is like because we all buy clothes and stuff like that or measure things in meters usually. So it's 1 millionth part of that meter. And if you look at this diagram over here, it's showing the brown thing is literally the diameter of the hair. And things like pollen grains, which a lot of you are, I saw looking under the microscopes, are much smaller than that diameter of the hair. So, when you collect those pollen grains, it's very hard to see each grain by naked eye because that's really, really tiny. And that's around 15 micron and you're able to see that. Yeah, Archana. Hi. You have a question? You're not, you're muted if you want to ask something. Okay, maybe she mistaken you. Okay, so the other things like neuron cell on the image or other cells are also very, very small. And to look at those scale, you have to really use some way to magnify it. So if you go to the next slide, we will later on talk about how we can magnify these tiny microorganisms or tiny things into bigger for our eyes to be able to see it. Again, going back to the classification of living system, as I talked about, there are different ways you can classify it. So one way to classify it is in a unicellular and a multicellular organism. So unicellular means one cell. So literally the organism is just one cell. So here is an example of paramecium on the top and algae also. So algae can be also in a unicellular form. It can be just one independent cell living happily. The other is the multicellular. So we are a multicellular organism where our whole body is made up of multiple cells, which forms this tissue and organ, and then our whole body. The other can be the plants as well. So plants are also made up of multiple cells stacked together. So as I said, they're like bricks stacking together and forming an organism. Again, algae can be in both. Algae can be a unicellular, it can be a multicellular. We will talk more in detail about that later on. The other way of classification of the living system, if we go to the next slide, is based on, I think, based on how their origin is. So for example, based on the origin, the classification works into a prokaryotic and a eukaryotic cell system. So prokaryotic cells are more archaic or were supposed to be one of the first ones which came to this earth in a way. And usually things like bacteria are classified in a prokaryotic cell. The major differences in prokaryotic cell is in terms of their DNA. So if you look at the eukaryotic cell on the right, it has a well-defined nucleus and it's a more sort of evolved system, whereas a prokaryotic on the left has just this nucleoid, doesn't have a well-formed nucleus inside. And again, it's just sort of how you think about these organisms. You can classify based on either the way they function or how their evolution happened. So if we go to the next one, the other way of the classification of the system is the plant cell or an animal cell. So we have animal cells, and of course the plants have the plant cell. And the major difference it has is in terms of cell wall. So plant cells have this really rigid cell wall where it's very nice for them to sort of, strengthen their body when the cells are stacking on each other. The other major difference is that plant cells can make their own food, right? So the way they do it is because they have these, they have chlorophyll and chloroplast, and we're going to talk more in detail about that later on. But again, just to keep in mind that that's another way to think about how we can classify living systems. Now coming back to the scale of life we were talking about, and what are microorganisms? Because I have been talking about that there are certain these organisms which are very hard to see by naked eye, and literally those organisms are the one which are microorganisms. So microorganisms are the ones which is microscopic in nature, which are usually in the scale of some micron. So if we move forward here, you will see in this in the scale, yeah. So the one in the red box is the one where we need some kind of instrument to magnify these microorganisms. So This will include all the things we talked about, like animal cell, plant cell, something like paramecium, bacteria, which are the prokaryotes. So these things we are not able to see by naked eyes. Things like yeast. So for example, the dahi you eat or the bread you make, these all have either some kind of bacteria or yeast in it, and it's very hard it's impossible for us to see it by naked eye. But one way to do that is to use an instrument to sort of magnify and to make our eyes be able to see those microorganisms. So again, microorganisms can be either good or bad in nature in a way. So there are harmful microorganisms. So for example, if you have, this thing like ringworm, which is very common in wet countries which have a lot of humidity. So that's also one of the harmful microorganisms that causes that ringworm. You can have these red algae and algal bloom, which we are going to talk about. So those are the examples of harmful microorganisms. However, a lot of microorganisms are really beneficial in nature. So for example, antibiotics are because of the because of the good use of microorganisms. As I said, like yeast in making bread is really important, or the bacteria in your gut, which keeps your gut happy, is really a beneficial microorganism example. So again, now to think about how we can magnify these microorganisms. Iqbal, if you would please go to the next slide. Please feel free to ask anything if I'm going too fast. If you have any kinds of questions as well in between, I'll be happy to stop. So what is a microscope? And I would like to maybe open the platform here and ask everybody, why would you use a microscope? And I already sort of gave you the answer, but I would also like to know maybe if you have some curiosity about why would you use one microscope, since you already have been using. And anybody?
And also, like, if you have actually used a microscope, like, just tell us, like, what all, things have you seen with it or whatever. Yeah.
It's an open platform, so please feel free to talk.
So are you asking about microscope versus foldscope, or you are talking about just a microscope?
Just a microscope. Why would you use a microscope? Or if so, foldscope is also a microscope.
Yeah, that's for sure, for to get the knowledge about the microorganisms which cannot be seen by our naked eyes, and to study these small pathogens or some disease causing elements, as well as the tiny structures of the plants, as well as the human bodies, as well as the animal things. So every kind of thing which cannot be seen by the naked eyes, and we want to study about it. So the microscope is the only solution for it.
Yeah, that's a perfect answer. You gave away pretty much what I spoke for last 10 minutes, and that's perfect. So how does a microscope work? Anybody has any ideas about that? Any clue? Ankit is nodding his head. Okay, so yeah, please go ahead.
So sorry. Oh, me again. It is basically it is the functioning of the lenses. We use lenses. Magnifying lenses are there, which have the ability to enlarge the tiny particles to show its clear or more large pictures. So microscope is basically works upon the physics of lenses.
Yeah, that's absolutely correct. So if Iqbal would go to the next slide. Thank you, Bhartiji. It's perfect answer. Yeah. So how does a lens magnify? As Bhartiji mentioned, you need a lens to sort of magnify whatever object you are looking at. So often the light from an object passes through a medium, and this medium can be lens, and it's bent, and so this bent is called refraction, and it can be refracted towards your eye, which makes the object look bigger. So here, I don't have the axis, so Iqbal, could you please show? So there is this angle of incidence, which sort of make your image look bigger. So if you look at the lens over here in the center, it is bending the light towards your eye. Because it's bending like this, the image in front looks much larger. Also, the way refraction works, it's very easy for us to have a look at home. So for example, if you take a glass, an empty glass, and put a spoon, you will see the spoon through the glass very well. But if you now put water in that glass, you will see that the spoon, or here you can see straw, it looks bent when you look through the water. That's because the light has to either go from the air to the water medium. So it's changing the medium. So on the left, when the light is going from air to this lens medium, it's undergoing this, it's making it bent, and that's the refraction there. So again, this is just an example of how a simple sort of magnifying glass would work. So the magnifying glasses usually have just one lenses, but microscope, Iqbal, if you move to the next slide, have often two lenses. So again, to compare between a simple magnification, by magnifying glass versus a microscope on the right. So the microscope has two lenses where there is an objective lens. So on the left, it's close to the object, whereas there is an eyepiece lens, which is often very close to the eye. The use of the objective lens is again to cause that bending of the light and actually magnify the image. So the first image is formed very close to the eyepiece lens, and then the eyepiece also magnifies it more and focuses the image on the eye. So it makes the image look much, much larger than it is. Iqbal, could you please go to the next slide? Is it Iqbal moving it or Kanika? It's you, right? Okay, sorry. So what are the parts of the microscope? If you keep moving forward, again, as I said, that there are objectives which are used for magnification. So there is the objective lens, which you will see close to the object stage over there. And then there is the eyepiece or the ocular, which is also used for more magnification. The other parts of your microscope is often the stage to support the sample, and then just to focus. So it's really important for those two lenses to be at a correct length to focus it properly. So the full scope, which you all have, has the exact same parts, except is just on a paper. So it has an eyepiece lens and an objective lens, which are very close to each other. The objective lens is sort of on the back side where you put your slides. So it's a little hard to see, but the eyepiece lens is right on the top over here. Again, you have this focusing part as well, which is made up of paper and stage. So I just wanted to show you that, even if it's a conventional light microscope or your foldscope, the components are very, very similar. It just looks slightly different. And if we move to the next slide, the way these two microscopes work, whether it's a foldscope or a conventional light microscope, is exactly based on the same principles, except it can have different magnifying power. So what do I mean by a magnifying power is how much larger a given lens can make an image appear. So it can have different kinds of lenses. And so a conventional light microscope can make an image look 40 to 1,000 times much bigger than it is. So if you have, let's say, have organism something around 100 microns, you can magnify it 1,000 times also in a conventional light microscope. Whereas the foldscope has a power to do it 140 times, which is already pretty powerful. So just to tell you that the instrument which you have actually is quite a powerful one to magnify and look at those microbes. Any other questions? Because I think I'm pretty much done with my part, and then I will hand it over to Kanika to talk more about algae and why we are doing this project, if there are no other questions. Kanika, take it over.
Okay. Thank you, Ankita. So, okay, let's make it, let's try to make it an interactive session, and feel free to interrupt us with any questions you have, in the middle of the conversation or whatever. But I just want to ask, how many people know about here algal blooms and algae, maybe algae in general, but more about algal blooms. Is everyone familiar with that concept or not?
Yes, ma'am.
Okay, awesome. So we were discussing with Ankita about the classification of different kinds of species. So our project is going to be focused around algae and cyanobacteria. And these are basically organisms which live in, marine or in freshwater environment. So these are aquatic organisms, and they can do photosynthesis. And, and these algae, again, in the bigger scheme of classification, so they can be eukaryotes, which means they can have like a well-defined nucleus, or they can be prokaryotes. I don't like to use the word prokaryotes, but they can be bacteria, which are mostly actually cyanobacteria. And these are the ones which are the harmful algae that we're going to discuss. And again, in eukaryotes, they can be either unicellular or they can be multicellular. So like unicellular algae would be like chlorella or diatoms, and multicellular would be something like more plant-like, which are like kelps. The interesting thing is so they're a little bit different from plants in the sense like they don't have, the roots or the stems or whatever, but they have the ability to photosynthesize. They can form these multicellular tissue kind of organs and everything. So I think we're all familiar with the concept of photosynthesis. So I don't want to stress here too much, but all of these algae, they can make their own food using this process of photosynthesis. And most of the algae, so there are some algae which are red in color, but most of the algae that, we are going to talk about, or maybe that you will also detect, will be green in color. And we're going to discuss this concept a little bit more when we discuss about spectroscopy. But it is a little bit important to remember that why these are green in color is because they have these chloroplasts, which are the sites where all the photosynthesis happen. And these chloroplasts have a green pigment called chlorophyll. And the reason it is green is because these chlorophylls, basically, they can absorb the blue and the red light in the visible spectrum, and they reflect the green light. So, like the sky is blue because the sky reflects that color. So everything, the color is basically a property of whatever the colors are reflected and not absorbed. And this concept we'll discuss a little bit more when we do spectroscopy, because it will be useful to measure the abundance. So, you can think about it as that, like if something is like in a very general way, like if something is more green, there is more abundance of that species. So we'll come to that concept back in spectroscopy, but it's going to be very important there. So just keep that in mind for now. And the reason why these algae are very important is because, like the first sort of oxidation event, like the major oxidation event which happened on Earth about 3.5 billion years ago, was due to the cyanobacteria, or Prochlorococcus. So these were some of the either these blue-green algae or the cyanobacteria that were there on the Earth originally 3.5 billion years ago. And in the process of photosynthesis, oxygen was made, and at a certain point when the oxygen levels exceeded, there was life which was able to live on oxygen, and that's why we are there. So that's why these species are very, very important, and they help to maintain the biodiversity. But as we know, excess of everything can be bad, and we're going to talk a little bit more about if you have a lot of algae, and especially algae which are very harmful, some of them can produce toxic compounds. It can be harmful, and that event, which we call as algal bloom, it's a very environmentally it impacts the environmental in a very negative way. So I see Iqbal has asked a question. How many of you have seen algal blooms in your neighborhood? So does anybody want to take a little bit of a pause and tell us, like, if you have seen algal blooms near your area, in any lakes or ponds?
Yes, ma'am.
You have? Where is
Yeah.
it, Survi? Can you tell us a little bit more, please?
मैम जैसे हमारे
Okay, first address me as Kanika. No, ma'am, please.
ओके कनिका, हमारे स्कूल्स में जैसे हमारे 8th क्लास में भी माइक्रो ऑर्गेनिज्म के बारे में बताया है, तो हम प्रैक्टिकली उन्हें दिखाते भी हैं, क्योंकि उनके गांव में जो वाटर बॉडीज हैं, मतलब हमारे स्कूल के पास भी ग्रीन कलर की एल्गी या फंजाई वगैरह हम उनको प्रैक्टिकली दिखाते हैं मैम। तो ऑब्वियसली जो बायो फील्ड से हैं तो सभी मे भी जानते ही होंगे कि एल्गी, और उन्होंने देखी भी होगी प्रैक्टिकली, और वो अपने स्टूडेंट्स को बताते भी हैं मैम।
So the reason I ask is like, anybody else wants to speak?
यस यस देवेंद्र कुमार, हमारे यहाँ पर भी जो स्कूल है उसके आसपास गांव है, उसमें पॉन्ड्स हैं, लेक, जो बिग पॉन्ड्स हैं, उनमें भी जो है जो यूनिसेलुलर एंड मल्टी सेलुलर एल्गी है वो बहुत ब्लूम करती है, और पूरा का पूरा जो वाटर है वो मतलब खराब हो जाता है, उससे पॉल्यूशन हो जाता है उसमें, वाटर पॉल्यूशन, और जो उसमें जो एनिमल्स हैं जो उनको भी हार्मफुल इफेक्ट पड़ता है।
Yeah, this, like, the reason I ask is that like ज़्यादातर जो algal blooms जो देखे गए हैं which are harmful in nature are actually near the coasts and everything, क्योंकि वहाँ पे there is like marine environment, salty, not like more sort of freshwater body, a marine environment. There is like sort of like what kind of species thrive in different kinds of environment depending on the salt content and everything. But ज़्यादातर harmful algal blooms, जैसे आप इस map में भी if you'll see, like the concentration in red is the highest here. तो यहाँ पे ऐसे near the coast ज़्यादातर red होते हैं. So it's very surprising that you also see this in inland areas. And this is actually one of the major aims of our project also, कि हम ये जो freshwater bodies है, lakes वगैरह, which are not near the coast but which are more inland, वहाँ पे किस तरह की algal bloom हो रही है. Iqbal has raised his hand. Iqbal, go ahead.
Yeah, I have a quick question for everyone. किसी ने red algal bloom या कोई और कलर का algal bloom देखा है pond या lake में? Because मैं जब मतलब छोटा था तो मैंने लाल रंग का, जो राइट हैंड साइड में ब्लूम है, वैसा मैंने बहुत देखा है. तो आप लोग के एरिया में यूजुअली ग्रीन एल्गल ब्लूम होता है या और कोई कलर के भी होते हैं?
यस सर, नमस्कार सर, मैं अनिल कमल भाटी. एक्चुअली सर, जब हम गुलावटी रोड पे मूव करते हैं तो ये मैंने ऑरेंज कलर का, ऑरेंज और रेडिश जो है एल्गल ब्लूम देखा था सर. मोस्टली अधिकतर हमारे एरिया में ग्रीन अवेलेबल है, बट गुलावटी रोड पे नहर है, उसी के पास में एक पॉन्ड है, वहां पर है सर इस तरह का.
हाँ, ओके, थैंक यू.
मोस्टली सर रेड और ग्रीन देखने को मिलता है, ब्लू बहुत कम देखने को मिलता है हमारे एरिया में.
Yeah, blue, I think, I don't even know, like, actually, like blue-green hota hai, which is like more, but exactly blue would be, I don't know, very difficult.
Exact blue nahi
Actually, I'm going to show you a picture. I'll show you.
Blue is not there. Blue will be not there because algae, the eutrophication we can see only through the green alga or the red ones, and which are very rarely in these areas, because we commonly found the green alga causing eutrophication in ponds and in water bodies, because in particular our village areas where our school are located, they are inland bodies. Inland water bodies are mainly the ponds, that no lakes are there, no rivers are there. So we can easily find out, because these are very contaminated due to the local pollution as well as the throwing of substances in the water bodies. So these small areas are having the green alga more, I think.
Okay.
Yeah, that is actually very, very interesting. मैं जो बोल रही थी, मैं एक पिक्चर दिखाऊंगी जब वो रात में मुंबई ने किसी ने पिक्चर्स ली थी, and they were like, you'll see blue sparkling color के. So, but you're very right. Like most of them are green and some red, which are very rare to find. And it'll be very interesting, I think, in the project, like as you guys take pictures with your students and everything, especially to see, like, because inland, like is people are looking at everything in the coast, but nobody's, like, very few people are checking ki inland water bodies mein kya ho raha in terms of algal bloom. So, so coming back to this thing ki algal blooms, ज्यादातर कब होती है? मोस्ट ऑफ द टाइम ये समर्स में ज्यादा प्रेवलेंट होती है, सीजन्स ऑफ अगस्त, सितंबर, जून, जुलाई टाइप, जब टेम्परेचर्स बहुत ज्यादा होते हैं. सो अगर आपने कहीं नोटिस पड़ा होगा, एटलीस्ट यहाँ पे जहाँ पे मैं रहती हूँ, कैलिफोर्निया कोस्ट के पास है, क्योंकि देयर आर मेनी लेक्स एंड ऑल इफ यू कीप गोइंग नियर द कोस्ट, जहाँ पे समर्स में नोटिस आ जाता है कि देयर इज algal bloom, so don't go near that water body. And these are some of the pictures, जैसे ओहायो यहाँ पे एक एरिया है, और फ्लोरिडा जहाँ, which are again like कुछ coast कुछ inland, but you can see these soup, एक green तरह का soup है basically, which they start seeing in these areas and where harmful algal bloom is detected. Or this is basically a very nice imagery near the Indian Ocean, so the Arabian Sea basically. और यहाँ पे आप देख रहे हो near the coast, so these are the areas जहाँ पे एकदम bloom है, algal blooms. and the satellite imagery, and you can see in the coast, yeah, inland maybe, and all near the coast, it's like heavy algal bloom in these areas. So actually, many algae are not harmful. So some of them are not toxic, especially which have these appearance of plant-like systems. So if you see like ऐसे rooted plants दिख रहे kelp की तरह हो या कुछ ऐसे, so plant-like algae या इस तरह के floating plants. So these are not harmful because I mean they're harmful maybe for the entire ecosystem, but they're not toxic is what I meant to say. So they don't produce toxic compounds as such. But the major algae which produce lots of toxic compounds, these are actually cyanobacteria. So these are prokaryotes, and they're mostly, they're single cell, but they have the ability to form multicellular colonies and filaments and everything. So if you see anything near your water body, and we'll ask you, to take pictures of water bodies near you, जहाँ पे आप ये project करोगे, ताकि हमें पता चले कि किस तरह की वहाँ पे algal bloom है. So it can be like ऐसे paint या soup की तरह हो सकती है, या कुछ ऐसे ये कटी हुई ग्रास इस तरह की आपको दिख सकती है, या ये स्कम which looks very horrible to look at, या इस तरह के ऐसे मैट्स की तरह अगर बिछी हुई है. So these are the kind of algal blooms which are very harmful, and we'll provide you instructions कि अगर safely water samples collect करने and everything, how to do that, because they do produce a lot of toxic compounds which can be harmful even to us people. So as we were discussing before, Abhi, just somebody said, I forget the name, sorry, but why do we have algal blooms? So one of the major reasons is because they get a lot of food, these bacteria, these harmful algal blooms. So a lot of nutrient runoff, excessive fertilizer. So ये सारा eutrophication जो हो रहा है कि हम इतना सारा, like if you are growing stuff and then हम उसके लिए कुछ भी plants वगैरह के लिए nutrients, fertilizers ये सब use कर रहे हैं. So all of it, like nitrogen, phosphorus, this is just getting washed off and into the water bodies, and this helps the algae to grow, like they are getting food. The other thing is climate change. So these, since they're photosynthesizing and they like warm temperatures, and that's why they're so prevalent in summers, क्योंकि जब गर्मियाँ शुरू हो जाती है तो उनको एक अच्छा temperature मिलता है, they come near the surface and they start growing. And obviously, like all these climate change and man-made activities are sort of interlinked. So there's a lot of correlation between that. Or so now I want to tell like some of the harmful effects of these algal blooms which are caused by cyanobacteria. Why are they harmful? Is because they produce these toxic compounds. जैसे कुछ अगर नाम सुने आप लोगों ने, microcystin, nodularin, anatoxin. Anatoxin is also called like a very fast death factor. So if you're exposed to बहुत ही small quantities of anatoxin also, it can be a cause of death or nervous system issues होते हैं. So like this was the image I showed, I was about to show you. This is near the coast of Mumbai. It's a picture which is taken in Juhu Beach sometime in 2018 or 2019. And you can see like a bioluminescence here. So this particular algae जो यहाँ पे आया था, this is called Noctiluca algae. And it is also called sea sparkle because you can see all these sparks in the sea and everything. And due to this, lots of fish were being killed on Mumbai in 2018, 2019. So it's I mean, it's very pretty to look at, but it is very, very harmful. And these are like some of the cases जो अभी US में यहाँ पे जहाँ पे हम रह रहे, like like 2021 August में there were like there were 3 dogs और वहाँ पे वो सिर्फ 20 minutes के लिए वहाँ पे swimming करने गए थे जहाँ पे इस तरह की bloom थी, and all 3 of them died within 20 minutes of being in the lake. So this or in 2014, 2007 में इस तरह के cases हुए थे चाइना में, ओहायो में या US में भी, जहाँ पे उनको बोल दिया गया था कि do not drink water because the water was so contaminated with algal bloom. So पूरे state को पूरे there was like a issue of do not drink water. Like you can imagine like the extent to which these can be harmful in that case. So just to summarize a little bit, because this is one thing that they can produce toxic compounds, but then what are the effects downstream? So what happens is that all this nutrient runoff is going into the system, which is causing more and more cyanobacteria to grow. So अब इसके बाद when algae die off, what happens is that they have to decompose. So all of this matter goes, for the decomposition, it just sinks to the bottom of the sea or the ocean or pond, wherever they are. And this decomposition uses up a lot of oxygen which is present. So basically all the other life forms which are there, like fish, they're being deprived of oxygen. And I just want to show you, like, these are some of the images which are taken like in US or in India. Like these are the, locations where harmful algal bloom has been detected. And most of them again is near the coast. And I think what is very exciting about our projects is that we are trying to target areas which are inland. So, in ponds and lakes and everything, and we will be able to see there. And this is something actually very interesting. We found out very recently, like last month ago, there is this, and we might be able to collaborate and do something together. Let's see. So there is this researcher, an Indian researcher in the University of Georgia, and he has found this thing which is called cyanocoj. So, like they're looking for harmful cyanobacteria, which is very exciting. And what they have done is that they have these images of any, kind of climate data or satellite, say, and they have like an online portal. So abhi in this portal, there are only 5 or 6 areas which are again near the coast, just Mumbai or Gujarat mein, or Tamil Nadu, which are near the coast. And they have like 5 or 6 places. And if you select, what water body you want to look at, you can also specify a time frame. So, you have data for summers, you have data for winters, and then you can see, just say, if you input, let's say from 2018 September to October, November, whatever, you can see okay, is time pe is particular water body mein, this was the chlorophyll content. So it gives you like a sort of a numeric metric measurement as well. And similarly, if you want to see what was the cyanobacteria density during this time in those particular water bodies, you also get a measure of that. So this is very interesting. And, depending on the kind of data you guys collect, we could also try to build something with them for our particular water bodies that we're interested in, like over time, how are things changing, and this is what the aim of the project really is, that we want you to actually continue this project for a year. And, every month, if you collect the data, you might be able to see changes in the kind of species, how much is their abundance and everything and so on. And yeah, like this was just the test areas that I wanted to show. Like they have some in Madhya Pradesh, Gujarat, Karnataka, Odisha, Tamil Nadu. So yeah, I mean, it'll be very exciting to see like more inland areas rather than just coast. Yeah, so this is the plan basically that we have. We want to measure what are the kind of species of algae that are present in a water body near you that you select, and what is their concentration. And like it's a global project in the sense we are also have another cohort which have already started collecting their data since November 2021. Some of them are in US, some of them are in other parts of India, in Telangana, and it'll be really good to have like more input points now. And it is a chance for us as well to be a responsible citizen, so we can see what's happening in our local environment. And to be able to, I just saw a message in the chat from Iqbal. Sorry, why is fish important to me? Because I'm like, it's not about you being a vegetarian, Iqbal. It is also about the entire ecosystem. For the fish are being killed, it's harmful. Sorry. And we will talk about more of this, about our website content later on. But we have created this repository called the Algal Web. It is actually from some other researcher, but he has imaged like hundreds and thousands of species of cyanobacteria, and we have images. So once you start taking data, we can also start analyzing those images. Ankit has a question. Ankit, go ahead. Ankit, you're muted.
Sorry. Yeah, I didn't want to cut you off. I had a small question. It might be silly. So do like water wells or groundwater have algal bloom or something like that?
I don't know, but I want to say no, since they're not exposed to like sunlight and everything, so they would.
But open water wells can be, right?
Open water wells definitely can, like scum or something. If you have seen, it's possible that there is algae in that.
But
groundwater, I'm a little bit skeptical of that just because they need
Got it.
sunlight, right? So yeah. Yeah, so these are like just some of the images from our present cohort. So you can see, these are some teachers who are again working with students in their respective schools. And they have taken some images like these green algae, some of them spirogyra and more of them. So we have been collecting this since November, and hopefully in the summers we'll probably see like all different kinds of species in whatever water bodies they're working on. And if we don't see, it's also a good thing, because this means at least the water body they're monitoring is not so bad. So either ways, I think it's a good practice for us to find out what are the good water bodies versus what are the not so good water bodies. So next time we are going to discuss what kind of safety measures to take when you're sampling the water in the field. What are the different kinds of algal species, the common ones that you will likely see, and how we can classify them. And we'll talk a little bit about spectroscopy, maybe that will happen probably in 2 workshops or so, and some more about collection of protocols for these collection of water samples for these protocols, how to image water samples and everything. And if people have imaged water samples, have they actually been, like when you had the workshop, did you learn how to like visualize water samples with the Foldscope? Anybody wants to say?
I think they did. I saw beautiful pictures on WhatsApp.
Oh wow, okay. I'm not on WhatsApp, so yeah. Sorry. Awesome. So yeah, we could do something like this. Like if you've already taken some images, we could discuss that as well, and if you have any questions, because you guys have been assembling Foldscopes, we could do that as well. I think Iqbal will probably say something at the end, but hopefully the way we would like to conduct the next workshop would be if you can have your Foldscopes assembled with you guys beforehand, and then we can do it together, like just some very basic sample preparation, stuff like that, together over Zoom, which is not the best platform for doing all these things, but in the given circumstances we can. And yeah, thank you everybody. So if you guys have questions, please feel free to ask us any questions.
I also wanted to ask if water bodies in Bulandshahr are used as, if in some parts used for drinking water or for other usages.
I'm not sure about all the water bodies, because I basically don't reside there. I have my school there. So I just think most of the water bodies are only using for throwing garbage, and some of the kettles maybe they took there to wash them, and not for the drinking purposes, at all.
That's good. Okay. No, I also wonder, people go for swimming and things like that in local lakes, and because if there are algal blooms, it's usually really harmful for that as well. Not just for fishing, Iqbal.
No, I asked that question because sometimes, I personally face that question that why do I care about fish kill? So I think we all must understand that every species that we see or don't see has some importance in the global ecosystem that we are in. So if you remove one species, then the consequences are unpredictable because it's a complex system. So you can never know, कि अगर पूरे इस food chain में से अगर हमने एक चूहे को हटा दिया तो क्या-क्या consequences होंगे. So it's not, but clearly bad consequences. Okay, so it is important to understand that despite your food preferences, we all must work together to take care of our environment. So my question to all of you is that, have you all started going to schools like during what's going on with the COVID situation? Are the schools open now, and have you started interacting with your students in person?
Yeah, schools are open now, and in fact, we are having the annual examinations of our students also. So it's offline now.
Okay, so that's great. ये अच्छी बात है कि आप लोग directly schools जा रहे हैं. And have any of you did you get a chance to show your students the tools that you have received?
यस सर,
Yeah,
हमने
of
अपने
course.
स्टूडेंट्स को काफी सैंपल कलेक्टिंग और ये सब दिखाया है, और पॉलन ग्रेन्स वगैरह, क्योंकि आप ग्रुप पे भी देख रहे होंगे सारे बच्चे
हाँ.
हमें काफी अच्छे से देख भी रहे हैं. अभी एग्जाम्स के बाद में जैसे ही हमारा अप्रैल से न्यू सेशन शुरू होगा तो बच्चे और एक्साइटेड रहेंगे ये सारी चीजें कलेक्ट करने में. और अब तो वो जो सैंपल कलेक्शन है ना, तो रोजाना मुझे एक बोतल थोड़ा सा सैंपल ला के देते हैं, मैम आज इस सैंपल को चेक करेंगे, मैम आज इस सैंपल को चेक करेंगे.
This is fantastic actually.
That's awesome.
Yeah. ठीक है, आई थिंक वी आर सुपर पम्प्ड कि आप लोग हमारे साथ जुड़े आज संडे मॉर्निंग को. एंड नेक्स्ट टाइम आई थिंक वी हैव मोर पीपल, राइट, कि ग्रुप में तो और भी लोग थे. सो आप बताएं अपने कोलीग्स को कि ज्वाइन करने के लिए, बिकॉज जितने सेशंस और आगे बढ़ते जाएंगे हम और डेप्थ में चले जाएंगे, जहाँ पे हम डिटेल्स बताएंगे सैंपल कलेक्शन रिलेटेड, डिवाइस यूसेज, डेटा अपलोड, डेटा कलेक्शन एंड वगैरह वगैरह. और हम चाहते हैं कि आप सब लोग कनेक्ट हो, इवन दो कुछ चीजें आप लोग के लिए बहुत बेसिक हो सकती है कि यार ये तो मुझे पता था पहले से, ये क्यों फिर से बता रहे हैं. बट ये सारी चीजें आई थिंक when you tie them together over multiple sessions, it will make sense and it will be very helpful for your students. Because at the end of the day, please try to make sure कि students experiments में involved रहें, और वो लोग भी समझे कि क्या डेटा कलेक्ट हो रहा है और ये डिवाइस कैसे काम करता है. हाँ, I see some faces.
जी प्लीज
बोलिए जोगेंद्र जी.
Yes, sir. Sir, one thing that I am concerning about, in our primary schools or in the area where we are going to teach our students, so how is it possible we can make aware them to about the algal bloom? So they don't know. I think basic. They don't know the basic idea of this algal bloom.
तो उसका सोल्यूशन ये है. जी जी, बहुत ही अच्छा क्वेश्चन है. उसका सोल्यूशन एक ये है कि ये जो स्लाइड्स और ये जो वीडियोस हम प्रोवाइड कर रहे हैं, ये आप डाउनलोड कर सकते हैं, ये यूज कर सकते हैं. अगर आपको ये स्लाइड्स चाहिए, आप हमें बताएं कि कैसे आप पढ़ाते हैं बच्चों को. अगर आप पेपर यूज करते हैं तो हम बेसिकली डॉक्यूमेंटेशन भी काम कर रहे हैं लोकल लैंग्वेजेस में, तो आप यूज कर सकते हैं स्टूडेंट्स को डिस्ट्रीब्यूट करने के लिए और समझाने के लिए.
Correct, sir. Actually, I am using all the possible ways of ICTs or projectors. So
we can give you slides. Will that be good for you?
You are saying yes. Sorry, ma'am.
नहीं, so वही जैसे I was just saying कि हम आपको या तो स्लाइड दे सकते हैं अगर आपको खुद पढ़ाना है, या अगर आपको वीडियोस.
मैम हमको कनेक्ट करना पड़ेगा. व्हाट आई वांट टू से, हमको कनेक्ट करना पड़ेगा उनकी बेसिक नीड्स से. फर्स्ट नीड इज द ड्रिंकिंग वाटर. तो हमें बच्चों को बताना पड़ेगा व्हाई वी आर द वाटर वी आर यूजिंग फॉर द ड्रिंकिंग पर्पस. इज इट सेफ और नॉट सेफ फॉर अस. वो पॉन्ड्स वगैरह ये सारी चीजें बच्चे कहाँ घूमते रहते हैं. तो कहीं ना कहीं मैं चाहता हूँ कि उस तरह के वीडियोज आपके पास हो या फिर पीपीटीज आपके पास हो, जिसके अगर नॉट ओनली द स्टूडेंट्स, हमें उनके पेरेंट्स को भी थोड़ा सा अवेयर करना पड़ेगा. देन वी कैन इन्वॉल्व देम इन दिस टाइप ऑफ प्रोजेक्ट.
पीपीटीज हम शेयर करेंगे और हम डॉक्यूमेंटेशन भी शेयर करने का सोच रहे हैं. लेकिन एक चीज आप सब लोग ये कर सकते हैं अगले बिफोर वी मीट, इज कि आप ये पता कर सकते हैं कि जितने स्टूडेंट्स हैं आपके ग्रुप में, बिकॉज एज फार एज आई नो आप सब लोग के साथ 5 से 10 स्टूडेंट्स होंगे, राइट, फॉर दिस प्रोजेक्ट, राइट, मिनिमम. तो जितने स्टूडेंट्स हैं आप लोग एक बार ये इंफॉर्मेशन कलेक्ट करिए कि वो सब स्टूडेंट्स का ड्रिंकिंग वाटर सोर्स क्या है. क्या वो लोग किसी लेक से डायरेक्टली पानी लाते हैं या उनके घर में.
सॉरी आई थिंक सॉरी टू इंटरप्ट यू. आई थिंक जहाँ तक बुलंदशहर पर्टिकुलर रीजन का इस एरिया का वो है, मोस्ट ऑफ देम यूजिंग द अंडरग्राउंड वाटर. आई डोंट थिंक कि उनको कोई लेक या बाकी अदर सोर्सेज जो है हमारे ड्रिंकिंग वहाँ से वो पानी यूज कर.
तो राइट,
वो भी जस्ट वो बोल रहे हैं कि जैसे खेलने चले गए या वैसा.
ओके.
हेलो, नमस्कार मैडम. मैं एक चीज शेयर करना चाहूंगा. मोस्टली जैसे हमारे बुलंदशहर एरिया में मोस्टली अंडरग्राउंड वाटर यूज तो करते हैं, बट इस चीज को भी इग्नोर नहीं किया जा सकता. मोस्टली अधिकतर लोग अपने डोमेस्टिक एनिमल्स को लेके वहीं जो नजदीकी पौंड और तालाब वगैरह वहां पहुंचते हैं, और जहां पर ये वाटर ब्लूम वगैरह, एल्गल ब्लूम वगैरह फैला हुआ है, वहां वो डोमेस्टिक एनिमल्स जाते हैं। देन उसके बाद में होता क्या है कि वो एल्गल ब्लूम्स उनकी बॉडी के ऊपर लगते हैं। तो ये एक ऐसा प्रोजेक्ट है जो उन लोगों को, जो बच्चे हैं उन लोगों को अवेयर करेगा इस प्रोजेक्ट के बारे में। हम ये नहीं कह सकते कि भाई वो वाटर बॉडी सिर्फ ओनली अंडरग्राउंड यूज कर रहे हैं, बट एक दूसरा भी एक पार्ट है जहाँ वो एक तरीके से आ जा रहे हैं, तो वो भी एक पार्ट है लाइफ का।
Yeah, that is actually a very good point. Yeah,
और
because
उस इंफॉर्मेशन से वो स्टूडेंट्स हैं या आप लोग हैं, ये अवेयरनेस भी फैला सकते हैं कि जो वाटर बॉडीज है उसमें एनिमल और ह्यूमन डिस्चार्ज ना डिस्पोज करा जाए, है ना। बिकॉज एल्गल ब्लूम होने का प्राइमरी रीजन ये है कि बहुत ज्यादा फर्टिलाइजर्स एंड ह्यूमन एंड एनिमल वेस्ट उसमें डंप किया जाता है।
एक क्वेश्चन मेरे लिए है, मतलब वो आई जस्ट वांट टू कंफर्म विद जोगिंदर जी अगेन, लाइक कि आपके लिए किस तरह का टीचिंग मटेरियल बेस्ट होगा, लाइक
आई थिंक मेरे लिए मैम वो टीचिंग, सॉरी,
वीडियोस या रिटन मटेरियल कैसा, मतलब
मैम आई थिंक मेरे लिए एक्चुअली आई एम ए साइंस ग्रेजुएट, तो मैं चीजों को जो आप कनेक्ट कर रहे हैं वो समझ सकता हूँ आसानी से। द पर्सन हु इज नॉट नो एनीथिंग अबाउट दिस एल्गी ब्लूम फंजाई पैथोजन है ना, एनी अदर थिंग, तो उनके लिए मुझे बेटर रहेगा कि एक वीडियो अगर फॉर्मेट में, क्योंकि हमारे यहाँ गाँव के बच्चे वीडियो फॉर्मेट में बहुत आसानी से समझते हैं, है ना, उनको इजीली इंटरेक्ट कर सकते हैं कि किस चीज के लिए हम इस चीज को यूज करना चाह रहे हैं।
ठीक है
ना मैम। तो वीडियो प्रोजेक्ट वो एक्चुअली पीपीटी स्लाइड्स आल्सो नॉट यूजफुल फॉर दिस पर्पस, क्योंकि वो समझ ही नहीं पाएंगे। पीपीटी में देखा, मैं पिक्चर्स देखा, देन वो स्कूल से जाने के बाद साइड में हो जाएगा, लेकिन वो वीडियो एक बहुत बड़ा इम्पैक्ट लेके आता है बच्चों के लिए।
ओके, ठीक है। हाँ, वो
थैंक यू सो मच। हम उस पे
वो
वर्क कर सकते हैं,
टीम में डिस्कस करेंगे। और एक्चुअली अगर किसी और को भी इनपुट देना है कि अगर आपको बच्चों को पढ़ाना है तो किस तरह का टीचिंग मटेरियल देना है, तो वो आप लाइक व्हाट्सएप या जैसे भी ग्रुप पे मैसेज करो हमें।
I also want to say something, ma'am, because
I think
most of the schools don't have most of the schools, ma'am, don't have the projectors or the LCD presentations there, because the electricity problem is very much common. So video is the thing. Okay, we can send it to the student, but we are not sure that every student will focus for the videos and they will go and check the videos. And we don't have that much of time in our regular time schedule, so that we can take out some time for this to show them the videos on mobiles. If you can provide any print rich material or the display material in the form of posters or in the form of any text, so that we can give it to the students to read them at their home, or we can paste them on the walls or in our offices where the students can get regular in touch with that material. It would also be a great help, because I don't think PPTs and videos are that much beneficial for my school circumstances.
हाँ, नहीं, ये बहुत अच्छी बात है। सबके अलग अलग रिक्वायरमेंट्स है। वो प्रिंटेड मटेरियल भी हम कर सकते हैं, लेकिन वो प्रिंटेड मटेरियल आपको प्रिंट आउट करना पड़ेगा। सो आई एम अस्यूमिंग कि आप वो कर सकते हैं।
नो इशू सर। आई थिंक भारती मैम आप सही कह रहे हैं। आई थिंक वन थिंग ये भी है कि हमें बच्चों से पहले उनके बच्चों को पेरेंट्स को भी गाइड करना पड़ेगा इन चीजों के लिए।
Okay.
I think और भी hands raised है,
गजेंद्र जी
and शशि जी।
कनिका जी, एक चीज और मैं कहना चाहूंगी, जो भी ये हम लोग के पास वीडियोस आप अवेलेबल कराएंगे, वो अगर हिंदी में हो तो ज्यादा बेटर रहेगा, क्योंकि वो लोकल्स हैं और वो लोग हिंदी ही समझते हैं।
परफेक्ट, वो भी कर सकते हैं जी।
वो भी हाँ, तो वो एक्चुअली आई थिंक एक आपके जितने जो भी इनपुट आ सकते हैं, फिर उसके बाद हम लोग आपस में एक बार टीम में डिसाइड करते हैं एटलीस्ट बेसिक लाइक आप फोल्ड स्कोप वगैरह तो जैसे आप उनको अपने तरीके से जब आप बताएंगे, बट कि एल्गल ब्लूम स्पेसिफिकली हम क्यों कर रहे हैं, उस पार्ट को लेके हम लोग कुछ थोड़ा क्रिएटिव सोच सकते हैं। वी कैन थिंक कैसे बेस्ट कर सकते हैं।
मेरा एक क्वेश्चन है, मैं गजेंद्र जी के बाद स्टार्ट करूंगा। तो
जी सर, मैं ये जानना चाहता हूँ, जैसे हम एल्गी ब्लूम पर मतलब शैवाल पे काम करेंगे, तो हमारा इसके पीछे उद्देश्य क्या है? जैसे हमने पता लगा लिया लगभग भारत की अधिकतर गांवों की, शहरों की अधिकतर जो पॉन्ड्स है, झील है, सब में शैवाल तो है ही, एल्गी ब्लूम है। मतलब इसके बाद क्या मिशन है कि गांव में क्या टारगेट रखा गया है कि हमने पता लगा लिया यहाँ पर ये ब्लूम है, एल्गी ब्लूम है, फिर उसके बाद हम कैसे, क्योंकि अभी तक हमने अपने स्कूल में केवल पॉलन पे काम किया है, बड़े रोचक रहे हैं, कुछ देखने के किस्से बच्चों को हमने दिखाया है। अपने व्हाट्सएप पर भी कुछ खास वो इमेज हमने शेयर की थी कि पोलन के बारे में, लेकिन अभी तक हमने अपनी केवल नल के पानी को इससे देखा तो उसमें कुछ कण नजर आए कि अलग टाइप के से, शायद उसका अभी हमें पता नहीं चला कि किस चीज के हैं, लेकिन उन्हें हम शेयर करेंगे तो पता लगाने की कोशिश करेंगे।
अब
पर इस
वाटर
सब
बॉटल भी
रिसर्च के
देखना
पीछे
बच्चों की, उसमें भी बहुत कुछ होता है।
हाँ, पर उसके पीछे जब हमें सब कुछ पता लग जाएगा तो हमारे इस मिशन का उद्देश्य क्या रहेगा?
Iqbal, you want to take that?
Yeah. Thanks, Gajendra ji, for the question. So, उद्देश्य मल्टीपल है। एक ये है कि किसी भी एरिया का जो ओवरऑल इकोसिस्टम है या बायोडाइवर्सिटी है, वो उसके वाटर बॉडीज और वाटर बॉडीज में रहने वाले जो जीव जंतु हैं उनके ऊपर डिपेंड करता है ना। तो उसमें फिश हो सकता है, उसमें माइक्रो ऑर्गेनिजम्स और उसमें मैक्रो ऑर्गेनिजम्स भी होते हैं। तो अगर हम ये इंफॉर्मेशन निकाल सके, एंड एल्गी एक कॉमन एलिमेंट है बिकॉज वो लोग फूड चेन के एकदम बेस में आते हैं, उनके ऊपर ही मोस्ट ऑफ द थिंग्स, उनको फिश खाता है, फिश को कोई और चीज खाती है, फिर पंछी, फिर जानवर, फिर ये वो। तो अगर हमने क्या किया कि हम लोग वाटर क्वालिटी को लेके बहुत इंटरेस्टेड हैं और हमने सोचा कि क्या एक ऐसा पैरामीटर है जिसको हम मॉनिटर कर सकते हैं ओवर टाइम, जिससे वाटर क्वालिटी का एक अंदाजा लग सके। तो हमने इसीलिए सोचा कि एल्गल ब्लूम या एल्गी इन जनरल इज अ गुड पैरामीटर जिसको हम मॉनिटर कर सकते हैं। और हम लोग टाई करना चाह रहे थे इस पूरे प्रोजेक्ट को साइंस एजुकेशन के साथ। तो इसीलिए हमने सोचा कि हम बच्चों को क्या सिखा सकते हैं एनवायरनमेंट के बारे में, उनको क्या टूल्स दे सकते हैं जिसको वो लोग यूज कर सके बेझिझक, बिना किसी रेस्पॉन्सिबिलिटी के। तो इसीलिए ये सारा प्रोजेक्ट है। तो इसमें एजुकेशन वाला कंपोनेंट तो है ही, लेकिन ये जो डेटा आएगा ये नॉट ओनली साइंटिफिक पर्पसेस के लिए अच्छा है बट ऑल्सो इन जनरल यूपी गवर्नमेंट के लिए भी अच्छा हो सकता है, लेकिन हमें नहीं पता। जैसे अभी यूपी गवर्नमेंट चाहती है कि वो सारे पूरे यूपी के जितने भी सोर्सेस ऑफ वाटर बॉडीज, वाटर सोर्सेस हैं, बॉडीज एंड लेक्स एंड रिवर्स, उनको मॉनिटर करे, लेकिन ऑब्वियसली वो पॉसिबल नहीं है बिकॉज ऑफ मैन पावर एंड सो मेनी इश्यूज, राइट। तो हमारा जो ये प्रोजेक्ट है ये एक नया पायलट है जिसमें हम कुछ चीजों को लेके एक्सपेरिमेंट कर रहे हैं। हम ये देख रहे हैं कि अगर हम सिटीजन्स को एंगेज कर सके इस प्रोसेस में तो ये प्रोसेस फुलफिल हो पाएगा कि नहीं। एंड दूसरी बात ये है कि आपके गवर्नमेंट ने, हमने ये बोला था एक बार कि सिटीजन द्वारा आया हुआ जो डेटा है वो इतना रिलायबल नहीं भी हो सकता है। तो ये आप लोगों के ऊपर है कि हम कितना अच्छे से ये प्रोजेक्ट करते हैं और कैसा डेटा हम लोग साथ में मिलकर एक्वायर करते हैं। तो
मैं जस्ट ऐड करना चाहती थी, जो हमारा मेन पर्पस है वो हमारा एज अ ओएफसी मेन पर्पस है इज आल्सो हम क्यूरियोसिटी को बहुत मतलब बेसिस पे रखते हैं। तो मेन एटलीस्ट हमारे लिए ये भी नहीं है कि सिर्फ डेटा कलेक्शन या वो हो, लेकिन बच्चों में चीजों की, जैसे आपने पोलन्स के बारे में बोला, उन्होंने देखा होगा, कभी नोटिस नहीं किया होगा, बट ऐसे होता है कि अगर आपको पता चल जाता है कि ये चीजें आपके आसपास हैं तो आप और चीजों को नोटिस करने लगते हैं। तो वो साइंटिफिक क्यूरियोसिटी बहुत उससे जागती है। तो ये एक बहुत बड़ा सॉर्ट ऑफ हमारा प्रोजेक्ट का बेसिस है, या हमारा इस पूरे ओएफसी का बेसिस है कि हम ये क्यूरियोसिटी को और इंक्रीज करना चाहते हैं। तो जैसे वाटर बॉडीज में आप बोल रहे थे, जैसे पोलन्स आपने इतने तरीके के देखे, वैसे आप एल्गी में देखेंगे कि इतने शेप्स के हैं, इतने अलग अलग यूनिसेल्यूलर है, मल्टी सेल्यूलर है, या उसमें वो मतलब बहुत अलग अलग वैरायटी दिखती है। तो उससे और क्यूरियोसिटी जागती कि क्यों है ऐसा, क्या रीजन है ऐसा।
मैं एक लास्ट पॉइंट ऐड करना चाहता हूँ इस पॉइंट पे कि अगर ये डेटा बहुत अच्छा हुआ, हमें कुछ अलग ही साइनोबैक्टीरिया मिल गया है, कुछ हुआ, तो ये सब चीजें साइंटिफिक पब्लिकेशंस में भी जाती हैं। सो वी ऑल एज अ ग्रुप कैन पब्लिश समथिंग जिसमें हर उस बच्चे का नाम, हर उस टीचर का नाम, जो भी इस प्रोजेक्ट में इन्वॉल्व है, जिससे वो डेटा आया, जिसने वो एनालिसिस किया है। तो ये ये अपने आप में एक बहुत बड़ी अचीवमेंट है, बच्चों के लिए भी, टीचर्स के लिए भी, और एक सिटीजन साइंस के लिए भी, जो हम प्रूव करना चाहते हैं that the data that we get from citizen science projects can be reliable.
Just adding to that कि last year actually they published एक नई species of cyanobacteria India में ही using a foldscope. Like उन्होंने ऐसे किसी एक नल में देखा था कि it was like copper के पास कुछ था, तो it was a very special kind of a cyanobacteria. So yeah.
I think
Shashi ji,
yeah.
Yes. Hello everyone. I had two things. First one is already discussed by Gajendra sir, and second thing is my own experience. I'm from Daryapur in Bulandshahr, so we had two ponds in Daryapur, and when जब बच्चे ने वो samples collect किए, तो we can compare that. हाँ, sorry. तो वो तो एक अमेजिंग एक्सपीरियंस था ये। सेकंड एक बहुत अच्छी सी बात बताती हूँ। हमारे स्कूल के जस्ट पास से कैनाल जा रही है, और हमारी स्कूल के बच्चे स्कूल से एब्सेंट होके कभी-कभी और स्कूल के बाद भी उसमें जंप करते हैं और खेलते हैं और नहाते हैं और मुंह में पानी एक एक्टिविटी करते हैं, पता नहीं अजीब सी एक्टिविटी करते हैं। तो मैंने एक दिन उनको पकड़ा और उनसे कैनाल का ही उन्हीं के द्वारा एक सैंपल मंगाया और उसमें माइक्रो ऑर्गेनिज्म उनको दिखाए, एंड तब उन्होंने वो देखे। मैम उस दिन से एक भी बच्चा मुझे उस कैनाल में नहीं दिखा।
I'm laughing.
नहीं, ये रियल लाइफ एक्सपीरियंस है मेरा, और वो बहुत यूनिक एक एक्टिविटी थी, और उसके दिन से बाद में मुझे एक भी बच्चा उसमें कैनाल में नहीं दिखा। और इवन उन्होंने क्लेज लिया, मैंने स्कूल में क्लेज दिलवाया बच्चों को कि अभी क्या करना है, हमें उसको डर्टी नहीं करना है। तो हमारी क्लास 5th तक के बच्चे जो है वो शाम के टाइम या जब अब आने वाला है नवरात्रि का त्योहार, तो उसमें क्या करते हैं, जो भी रिचुअल्स जो भी चीजें होती हैं उनको एक बहते पानी में थ्रो किया जाता है। लेकिन अब क्या कर रखा है बच्चों ने कि हम देखेंगे मैम कि कौन थ्रो करता है, कौन गंदा करता है। तो एक ये जो सिटीजन साइंस का एक बहुत अच्छा ये बेनिफिट है कि हमें अपनी वाटर बॉडी को गंदा नहीं करना है। तो ये कहीं ना कहीं छोटे बच्चों में डेवलप किया है और स्टार्टिंग हुई है कहीं ना कहीं से। तो देखेंगे अभी आगे आने वाले टाइम में कि हमारे यहाँ के जो पोंड है उनमें क्या रिएक्शन होता है। बाकी हमने जो 3 या 4 पोंड जो है हमारे यहाँ पे बाटगढ़ी में है, दरियापुर में है 2-3 पोंड, तो उनमें भी हमने कंपैरिजन किया है। हमने देखा है कि किस तरह का एल्गम ब्लूम और फाइटोप्लैंक्टन हमें मिले हैं। एक चीज है जो मैं डिस्कस करना चाहूंगी, बहुत सारे माइक्रो ऑर्गेनिज्म उसमें दिखाई देते हैं। मैं मैथ से हूँ, कहीं 10th में साइंस पढ़ा था, तो मैं उनको ना डिटेक्ट नहीं कर पाती हूँ और आइडेंटिफाई नहीं कर पा रही हूँ थ्रू फोल्डस्कोप। तो उसमें मुझे हेल्प चाहिए मैम।
हाँ, हम आपको
अंकिता,
पूरा
आर यू
ट्रेन।
एबल टू अंडरस्टैंड? हेलो, हाँ जी, एक बार बताइए।
हाँ शशि जी, थैंक यू सो मच। दिस इज लाइक आपने जो बोला, दिस इज द बेस्ट थिंग आई हैव हर्ड टुडे। मतलब बहुत ही इंस्पायरिंग और
थैंक
एक्साइटिंग है।
यू।
और हम लोग डेफिनेटली कोशिश करेंगे पूरा आपको और आपके स्टूडेंट्स को ट्रेन करने का। और आप ये भी ट्राई करिए कि वो जो स्टूडेंट्स हैं वो अपने मम्मी पापा और घर में भी जाके ट्राई वो वीडियोस या वो सारी चीजें दिखा पाएं कि उनके ड्रिंकिंग वाटर में क्या है, वो लोग जिस पानी में नहाते हैं उसमें क्या है। तो उससे जो अवेयरनेस फैलेगी पर्सनल हाइजीन का या इन जनरल टेकिंग केयर ऑफ योर वाटर बॉडी, वो बहुत ही अच्छा टेक अवे होगा इस प्रोजेक्ट का।
यस सर, एक्चुअली दे आर डूइंग। वो उन्होंने पता क्या बोला है घर जाके, उन्होंने ये नहीं बोला कि हमने इस वाटर में ये देखा, उन्होंने बोला हमने एक बूंद में ये देखा। They said we saw in one drop। तो वो 1 ड्रॉप वाला जो कॉन्सेप्ट हुआ ना, तो उनके जो भी पेरेंट्स थे, तो जो कम्युनिटी से आ रहे हैं, तो वो आए स्कूल में। और अब मैंने 4 हमारे पास जो है सीडीओ सर ने 5 किट और भिजवाई है स्कूल में इस तरह के प्रोजेक्ट को देख के। वो भी बहुत एक्साइटेड थे। तो जो किट है मैंने 4 से 5 फोल्डस्कोप रेडी कर रखे हैं, और मुझे हमेशा लगभग 2 दिन, एक दिन में 2 स्लाइड मुझे तैयार करनी होती है, क्योंकि कोई ना कोई आ जाता है कि एक बूंद में ऐसा क्या है। तो वो एक बूंद में मुझे उन्हें दिखाना होता है कि ये एक बूंद में ये है। बट समटाइम्स में बहुत डिफरेंट से माइक्रो ऑर्गेनिज्म उसमें तैरते नजर आते हैं, तो आई कांट एबल टू फाइंड देम कि ये क्या है। तो मुझे भी नहीं बता, मैं उनको ये बोलती है माइक्रो ऑर्गेनिज्म है।
हाँ, मतलब विल ट्राई टू मेक श्योर कि जो वीडियोस जो पिक्चर्स आप या खींचेंगे फोल्डस्कोप यूज करके, हम आइडेंटिफिकेशन करने की पूरी कोशिश करेंगे। तो हमारे पास एक पैनल ऑफ एक्सपर्ट्स हैं जिनका डेली जॉब है एल्गी और एल्गल ब्लूम ये सब पे रिसर्च करना। तो वो लोग डेफिनेटली आपको हेल्प करेंगे।
I also quickly wanted to add कि अभी तो WhatsApp में ऐसी for fun sort of share कर रहे हैं, उसके बाद सायक और अंकित बताएंगे कि कैसे एक जगह हम सारे डेटा अपलोड करेंगे। तो वो सारा जब एक साथ होगा तो और आइडेंटिफिकेशन इजी हो जाएगा। अभी sort of WhatsApp में हम बाकी लोगों से शेयर नहीं कर रहे हैं और जो एक्सपर्ट्स हैं उनके साथ। तो एक बार आगे के वर्कशॉप के बाद जब हम उसमें जाएंगे तो वो हम डिसाइड कर लेंगे कि कहाँ अपलोड करना है और उसके बाद फिर स्टार्ट करेंगे।
Thank you, experts. Thank you.
I think दो और क्वेश्चन है पूनम जी के
नेक्स्ट
and NK
इज
जी के
आई थिंक विल गो टू पूनम जी। डू यू हैव अ क्वेश्चन?
and NK जी के।
हेलो हेलो
Hi.
मैम, मैं नरोरा जो जहाँ पर गंगा नदी है वहाँ पर हम रहते हैं। तो जो गंगा नदी के किनारे जो है वो काफी मतलब थोड़ा गंदा तो बहुत ज्यादा रहता है, लोग बाग फालतू चीजें डालते थे, अब तो खैर बैन हो गया है और अब नहीं डालते हैं, लेकिन फिर भी काफी साइड में बहुत गंदगी रहती है और बहुत ज्यादा ऐसे गर्मी में भी काफी ग्रीनिश जैसा आप बता रहे हैं एल्गी वाला बहुत ज्यादा रहता है। तो और लोग पीते भी हैं पानी क्योंकि गंगा को अब भारत में तो बहुत ज्यादा मानते हैं, तो इसीलिए तो मैं सोच रही हूँ मैं वहीं से कलेक्ट करूंगी। और आप बताइए वहां पर रनिंग वाटर में कुछ मिल सकता है क्या?
जरूर। आई मीन ये क्वेश्चन मुझे पूछना था कि आप लोग सब लोग फ्रेश वाटर बॉडीज से तो लेंगे, बट कोई नदी के पास रहता है? तो ये बहुत ही अच्छा आपने ऑलरेडी आंसर कर दिया ये चीज कि नदी से भी लीजिए, लेकिन नदी सिंस वो रनिंग वाटर है उसमें ऐसे स्टेग्नेशन नहीं होगा एल्गी का, बट उसमें डेफिनेटली एल्गी होता है। लेकिन क्वेश्चन ये कि आप किस साइड से लेंगे और आपको थोड़ा सावधानी भी रखना पड़ेगा।
हाँ, क्योंकि जनरली जहाँ पे एल्गी होते हैं वो वाटर थोड़ा सा बेसिकली ठहरे हुए वाटर में एल्गी की ग्रोथ ज्यादा होती है।
So I
तो
think
अगर हाँ
थोड़ा सा क्लाउडी होगा, बट ये बहुत ही अच्छा डेटा पॉइंट होगा जस्ट टू सी कि उसमें क्या क्या है, किस टाइप के माइक्रो
यूजुअली
ऑर्गेनिज्म।
गंगा में जहाँ पे बोट्स या जो शिप तो नहीं होती, वहाँ पे बोट्स ही होती है, वो पार्क्ड होती है जहाँ पे, वहाँ पे आई थिंक वाटर बहुत स्टैग्नेंट रहता है। जो भी कोई भी आके कोई रिचुअल भी करता है तो उसी एरिया में करता है। तो आई थिंक वहाँ पे algal bloom और even different kind of bacteria मिलना बहुत पॉसिबल है।
जी हाँ, जी 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 सर।
ओके, नेक्स्ट है NKB। मुझे फुल नेम नहीं पता।
नमस्कार सर, मैं सर नीलकमल भाटी।
अच्छा,
मैं दो पार्ट कवर करना चाहूंगा सर। जैसे कि अर्लियर स्टेज में अभी हमारे किसी साथी ने डिस्कस किया था कि जैसे पीपीटी और वीडियोस हमें अवेलेबल कराई जाए। तो मोस्टली हम एमएससी हैं, मोस्टली अधिकतर बॉटनी से भी होंगे, तो पीपीटी और ये वीडियोस वगैरह हमारे लिए तो अच्छा है, बट मोस्टली हमें करना क्या होगा कि बच्चों को जो फ्रेश वाटर है प्लस जो पॉन्ड का इकोसिस्टम वगैरह है वो बच्चों को वहां ले जाकर कंपेयर करके समझाना पड़ेगा। तो एक समझ बूझ पैदा होगी बच्चों के अंदर, वो अपने आंखों से देखेंगे कि फ्रेश वाटर क्या है और जो पॉन्ड इकोसिस्टम के अंदर जो माइक्रो ऑर्गेनिज्म मिल रहे हैं वो किस तरह के हैं। तो एक समझ जब यहाँ से पैदा होगी तो बच्चे के अंदर क्यूरियोसिटी डेवलप होगी, तो हमारा पहला काम वहां से पूरा हो जाएगा कि पीपीटी वीडियोस के थ्रू हम ट्रेंड होंगे और उसके कंपैरिजन के थ्रू हम बच्चों को वो बारीकियां सिखाएंगे। देन अगर दूसरे पार्ट पे आए, जैसे कि गजेंद्र सर ने कहा था कि आखिर इस प्रोजेक्ट का प्रोस्पेक्टस क्या है या क्या है इसका टारगेट, तो हम एक चीज पहले जाने कि जैसे हम पहले HAB के बारे में बताएंगे बच्चों को कि हार्मफुल एल्गल ब्लूम आखिर है क्या चीज, वो किस तरीके से इफेक्ट करता है, क्या वो डिजीज पैदा कर रहा है। तो हम इस चीज के अंदर सबसे पहले तो वाटर डिजीज के बारे में बच्चों को बता सकते हैं, इन चीजों से क्या ऐसी डिजीज हमारी बॉडी के अंदर फैल रही है। देन वन ऑफ द मोस्ट इंपॉर्टेंट थिंग इज दैट फूड चेन एंड फूड वेब, ये जो बहुत जरूरी है कि हमारे इकोसिस्टम पूरा फूड चेन और वेब से इंटरकनेक्टेड है कि किस तरीके से एक ऑर्गेनिज्म अगर मरता है इस लाइनअप में से तो वो किस तरीके से हमारी पूरी होल लाइफ को डिस्टर्ब कर सकता है। जब तक बच्चों के अंदर ये सारी चीजें आ जाएंगी देन वो इंटरेस्ट लेंगे, और ये हमारी अर्थ के लिए भी एक अच्छा प्रोजेक्ट है, क्योंकि एक स्पीसीज भी अगर इफेक्ट होती है देन इनडायरेक्टली हम भी उस चीज से इफेक्ट होते हैं। थैंक यू।
आपने बहुत अच्छे से समराइज कर दिया हमारे पूरे प्रोजेक्ट को, शुक्रिया। एंड यही है बेसिकली जो अंकिता ने भी बोला। आई जस्ट वांट टू से कि अगर इस प्रोजेक्ट के बाद हर स्टूडेंट या उनके माता पिता या कोई भी जो उनके कांटेक्ट में आता है, जो इस प्रोजेक्ट के कांटेक्ट में आता है, वो थोड़ा सा भी अपने एनवायरनमेंट को लेके थोड़ा सा भी ज्यादा क्यूरियस हो सके तो ये हमारे लिए एक जीत होगी। ओके, नेक्स्ट क्वेश्चन नहीं है। आई थिंक वी आर ऑल सेट, सबको सब समझ में आ गया आई थिंक। ओके, अगर कोई क्वेश्चन नहीं है देन आई थिंक दैट्स इट। वी विल मीट नेक्स्ट टाइम। सो नेक्स्ट वीक का डेट